All right, we've gotten past the technical issues, so everybody knows Dr. Prouty. Dr. Prouty is a cytogeneticist and has been in the pediatric department for a number of years running the cytogenetics lab. And he's sort of been in the middle of this evolution or maybe revolution in genetic testing that has occurred in the past 20 or so years. So they ran rounds with some molecular testing.
Thank you, Joe. Okay. Well, I have nothing to declare. You're either now ordering or will be ordering very soon tests which have a molecular basis. So I thought I would take the opportunity to get you into these testings, show you something about how they work so you can sort of expand your mental menu of the tests and perhaps even use them more selectively. My major recollection as a new cytogeneticist here some years ago was a resident coming up bringing a tube of sodium heparinized peripheral blood and asking me if I could do the chromosomes on it to find out whether the person had the defective allele for cystic fibrosis. I should have been complimented, I guess, but of course the cytogenetic test is not the appropriate test. That does not have the adequate resolution for looking at things at the base pair level. So I want to get you so that you don't have any embarrassing moments like that, certainly, and also to inform you of some of the new developments that we're coming up to. Now, what do I mean when I talk about molecular? Well, we've been doing molecular testing for a long time, specifically with proteins. And the definition of molecular diagnostic testing includes proteins, DNA, and RNA from tissue or fluid. And so we've been doing this sort of thing for 75 years, I guess, electrophoresing hemoglobin, showing that normal hemoglobin B and a person heterozygous for the sickle cell allele and a normal allele. We've been identifying very strange bacteria and virus with immunological technology, and so we are fluorescing and lighting up this particular strain of E. coli and identifying it and not having to culture it out. So that was, a great advance. So in proteins now, we're entering a whole new world where we have this new technology, the multi-top spectrophotometry, and we can take a group of proteins embedded in a matrix here of various identities, and with the laser stimulation, we can eject these proteins from the matrix. They go down a column which identifies them, essentially, by their mass and their charge. And so this is a tremendous advance, very quick recognition of proteins, has already applications in microbiology and certainly also in genetics, clinical genetics. Imagine instead of just doing a heel stick and looking for a few diseases, for newborns, you could use this sort of technology, and you could find a range of abnormal and rare diseases so that you may not be able to do anything about the disease too much if it's a severe metabolic disease, but at least you won't have a situation of diagnosis and crisis when the baby comes to the emergency room. So here's potential there. But I'm not going to be talking about proteins. I'm going to be talking here about the DNA and the RNA side of things. So, we use these tests not just for diagnosis, but we monitor people, stage of disease, viral load, response to therapy, so forth and so on. So, and before we get into this, however, a little technical knowledge is needed, and the way I propose to do this is just to very quickly and superficially go through the technical development of the techniques that we now use and the times they were discovered. So back in 1958, when we, just 5 years after the publication of Watson and Crick's paper, we discovered the DNA polymerases. The things that are responsible for replicating DNA and transcribing DNA from single-stranded forms. We also, shortly thereafter, discovered a very important enzyme, reverse transcriptase, which is able to take an RNA template and produce DNA from that. Has meaning later on. In the 1960s, the early 1960s, we really couldn't do much with DNA. We could chop it up a little bit with enzymes which were rather indiscriminate. They'd chop any particle anywhere along the double helix or eat away at the ends of it. We were able to fractionate the DNA, however, of a certain species, say human, and denature it, which is to heat it so that the double strands come apart, and then renature it. And we did a number of experiments in kinetics showing that, well, some segments of DNA renatured very quickly, and some sections of DNA didn't reanneal very quickly at all, didn't renature quickly. And that just corresponded to the first group being repetitive DNA, non-coding DNA such as we have in the centromeres of our chromosomes, and the long to slow to renature DNA with very particular pieces of DNA, unique pieces of DNA that, for example, made up our genes. So we learned very on that DNA had differences in it. Okay. Then in 1970, the discovery of the restriction enzymes. These are enzymes in bacteria which can cut the DNA at very specific sequences. For example, there's a 5 base pair sequence here for the EcoR1 restriction endonuclease, and it cuts it just at this point, leaving these little overhanging edges. And it has to be just this sequence for it to act. The scads of other restriction enzymes that we discovered. So for the first time, we had the ability to cut the DNA in very precise ways. Then, by 1973, we used this ability of the restriction enzymes to isolate a gene, stick it into a bacterial plasmid, which is a little circular DNA molecule, usually which carries the plans for the antibiotic resistance for the bacteria, put that back into the bacteria, grow the bacteria up into millions and millions and millions of copies, then lyse the bacteria, extract the plasmid, and then cut the little gene segment out from the plasmid. This means we've got millions and millions and millions of copies of that gene. So we have cloned a gene, and that is that went back to 1973, a tremendous achievement. Then by 1975, we took these cloned genes that we had, and we did a little bit more with the restriction enzymes. You know, you might have 3 patients here. We took their DNA, we chewed it up with a particular enzyme, a particular restriction enzyme, put their DNA on a gel, and then transferred the results of that gel to a nylon membrane so that we had all the single-stranded sequences in the order in which they had migrated in the gel. And then because we have a probe for a particular gene, we could make it radioactive and just see if we could get that probe, make it single-stranded, and see if it'll hybridize to this membrane. And it will in different places. And what this means is that this particular enzyme has cut that segment of DNA in 3 different ways. It's the same segment of DNA. These are 3 different people, but we've got a little variety here in the base sequences of these people, because the enzyme can cut in one place, in one person, which has that restriction site, but in the same locus on the next person, there's been a base pair change, like a point mutation. So the enzyme skips that, and the fragment that results is longer. Okay, so just some discriminations there that were possible. Now, for the isolation of cDNA, that came in 1976. Very important. cDNA is simply DNA that is based on the mRNA that you extract from cells. So here we've got the mRNA in a cell, isolate that mRNA, and we use this special enzyme reverse transcriptase to take the mRNA, the messenger in the cell, and then, which must be hundreds of them inside a living cell, hundreds of different ones, and then we make a whole library of these things, but we put them into DNA form, okay? And so this is very important. What we have done is we have made DNA clones, because we can amplify these in our bacterial system. We have made DNA clones of active genes by simply raiding the mRNA that's in the cell. So that was very clever. Now, what these clones are and what genes they represent takes another step of research. But those answers could also be gotten. Now, then by 1977, we'd long had the ability to sequence proteins. Now, at last, we have the ability to sequence DNA. And we set up 4 different reaction vessels in this Sanger sequencing context. And each has one of the bases, adenine, guanine, cytosine, or thymine, and the enzyme that's necessary to do the replication. And, of course, the sequence that we want to know the base pair sequencing of, we'll throw that in too. But we also put in separately into each of these vials, a nucleotide that's a little different. It's marked, now we do it with fluorescence, and these are the dideoxynucleotides. So what we do is we just say, go to it, start making your DNA strands on this template. But if we're starting here, and this is the complementary nucleotide to that adenine here, and it's a dideoxynucleotide, our little strand stops here. If we go all the way through the process and we stop the reaction here with a dideoxyguanine, okay, this will stop this reaction here. Everything in between represents lengths of DNA where the reaction has stopped because of the dideoxynucleotide. Okay, so these are of different length, and we'll just separate them on a gel so that the shortest one comes out down here, the longest one is up here. Everything in between is represented, and that tells us what the sequences of bases have been in that particular sequence. Okay? So this is the gold standard. You can get pretty long results from this. We still do Sanger sequencing, and it's what we started off doing when we were doing the Human Genome Project. We figured out we couldn't do it fast enough with this. You can only use one clone at a time, one stretch of DNA at a time when you're doing your sequencing for Sanger sequencing. But we've got sequencing now. Okay, and then we go along and we create something new called the oligonucleotide sequence. I won't go too much into the particular chemistry of this. It's the ability to create short nucleotide segments, usually about 20 nucleotides long. Well, and we can do this in specific reaction chambers. We don't use aqueous chemistry and stuff like that. Very clever. If you consider that there are 4 nucleotides, and we shoot for a segment of these nucleotides that's 20 base pairs long, this is the number of combinations that we can come up with. That's very important, because if we want to use these little pieces of DNA to go back to genomic single-stranded DNA and get a gene to be expressed, these can be the primers for the expression. Remember that the polymerase that's going to make the DNA has got to sit down on a double-stranded stretch of DNA. It likes to add bases to a single-stranded piece of DNA, but first it has to find a double-stranded piece of DNA, and that we can get to by using these little oligonucleotides. We can make an oligonucleotide that can be a primer for any gene in the genome, and that's a big help. Okay, now, so finally we got our act together here with our southern blots, and we're able to use our restriction enzyme technique and our electrophoretic techniques to label particular, characterize particular genes. For example, we can have a genotype. This is our restriction enzymes give us something, a segment of DNA that migrates in this way. If it was heterozygous, this person would have a shorter piece and a longer piece, and if it's homozygous recessive, we just have the shorter piece. Okay, so let's compare our family here, which has a recessive disease segregating in it. This is an affected member here, and this is what his DNA pattern looks like from a restriction enzyme southern blot type context. And the normal people, carriers, look this way, and here's a person who isn't a carrier and is normal. So, okay, so that's a new way of doing things. Now, the polymerase chain reaction is sort of very much of a standard in our armamentarium here, and it's based on the fact that there are these peculiar aquatic bacteria that live in these very hot pools like you have at Yellowstone, and they have a polymerase which is able to work at 72 degrees centigrade. So what you do is you have a segment of DNA of interest that you want to amplify. You heat that up in your reaction vessel to 95 degrees. The strands denature. You have primers to start your replication. You're using the Taq polymerase here, and you put your primers on at 55 degrees. They'll go on at 55, but you won't get a re-annealing, a mass re-annealing of the whole segment of interest at that temperature. And finally, you'll then just make the complementary strand. You just go through the cycle over and over again, and you can produce as many copies of your segment of interest as you want. The great advantage to this is that we can create clones of DNA, but we don't have to do this business of putting it into bacterial plasmids and raising up the plasmids and all of that messy business. We can do it directly in reaction vessels on the bench top and get tremendous results as a result. Okay, about 1986, we're starting to do FISH, which is a cytogenetic molecular technique. We have probes now. We have clones of DNA from a certain part of the chromosome, a certain part of the genome. We can label these probes with fluorescent dyes. We denature the chromosomes and the chromosomal spreads on our microscope slide, and then this probe will simply go back to where it came from on the chromosome, and there we have it. If that segment, for example, however, was missing due to a deletion on that chromosome, you'd only see one red dot here showing the probe settling in. There'd be no place for that probe to settle on another chromosome. So this chromosome would be normal, show two red dots. If you had another chromosome, its homolog, which didn't have any dots on it, that would indicate that there had been a deletion. Okay, works that way. Now, so then we get into real-time, a quantitative PCR. So we've got a PCR reaction going between two individuals, sample A and sample B, all right? And a PCR reaction goes along at a slow pace to begin with, okay? If you could visualize the reaction going forward, it would look something like this. You wouldn't really see anything much. It would look all rather quiet. And then suddenly get to a point where mathematically this reaction takes off and it goes into its exponential phase. In other words, you're getting noticeably larger amounts of the DNA amplified, all right? So, and this is the exponential phase over here. Finally, it plateaus off because at the end of the reaction, all the reagents are used up, okay? So, where this thing takes off here is an indication of how much of the sample was there to begin with. In other words, how many of this little segment that we were going to amplify was in the reaction vessel to begin with. If there was a whole lot of it, you're going to see this curve going up very fast, very soon, after only a certain number of PCR cycles. If it was just a very small amount, it takes more PCR cycles to amplify that specimen up to the point where you can actually see it. Well, how do you see this happening? Well, there's a very clever way of doing it. If you have your segment here, in which you've got your probe, this is the segment that you want to amplify here. There is a probe that can go into it, a little oligonucleotide that has a fluorescing reporter molecule on it, but a quencher molecule very close to that. So as long as these two, they're going to sit down right in the middle of this sequence here that you're going to amplify, but you can't see the fluorescence because the quencher is covering it. As the amplification goes on, however, and the enzyme reaches this point, it knocks off the fluorescing molecule, releasing it from its quencher, and this is seen, okay? So when this is seen, the PCR machine is looking for it. And it looks for fluorescence and what quantity the fluorescence is occurring to as an indication of the stage of the reaction. Is it in exponential phase or is it not in exponential phase and so forth? So it can read the progress of the reaction by the release of these fluorescing molecules. Okay, so that's how we can get these curves here and know where they're going. It's called the TaqMan system. It's based on Pac-Man of the computer game. Okay, now by 96, we're doing microarrays. And so we can learn all sorts of things from microarrays and the three basic different kinds of microarrays that I want to get you familiar with. So the way they work is that you get, you can take these little oligo clones, okay, and you attach them to the surface of some kind, glass, silicon, whatever, in an orderly manner. You form thousands, perhaps millions of spots of these on your substrate. Okay, this is like a cassette that has this in it. And each one of these little spots represents something that you're looking for. A segment along a chromosome, for example, if you're doing a chromosomal array. It could be a very particular part of a gene, all right? A gene that, a part that has a common mutation in it. So you can arrange these very rationally in an array fashion, and you label these things with a fluorescent dye, and so these are our probes. In a fish probe, the fish probe is in solution and it attacks a target, which is on the microscope slide, which is the chromosome. In this case, it's the other way around. We've got our probe sitting here already attached, and we're going to take our patient sample, and we're going to label it in a complementary way, we're going to PCR up just the kind of portions that will respond to these probes out of the patient sample, and we're going to then try to hybridize the patient to the, which is the patient, which is the target to these probes. Okay, now if you get hybridization, if you get the patient material, which is labeled in green, to hybridize at one of these places here, just a red signal, a green plus a red signal produces a yellow signal through the microscope, which is looking at that position and recording what's going on. Because these microarrays are just amazing. They're totally automated, and so each one of these little positions will be looked at by a microscope, bump, 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 and the information recorded in a computer, and that's how we know what's going on, what's reacting to what. Now, why that's yellow, I can't tell you. It has to do with the physics of fluorescing molecules, but that's the way it is. Okay. Now, so we can have all sorts of scales of detail for our microarrays. We can look at individual point mutations by looking at what we call now SNPs, which just means a single nucleotide polymorphism. Just a place in a segment of interest, a gene or what have you, where instead of having AT at this site, we have a GC pair and vice versa, okay? So that can be very precise. And we can see also small deletions and duplications that way. On a larger scale, we can look for large missing pieces, like the sort of things that we might want to look for in a chromosome study. So, but we can do that with array technology, and we can do it with much greater resolution. So here we have, you know, a match-mismatch type situation, hybridization producing a yellow signal, and no hybridization for the SNP microarray producing a red signal here, because we didn't get the greens to really combine. All right, so that's how the microscope tells the difference. There are all sorts of controls that we involve in this. I won't go into this. But another kind of microarray is a gene expression microarray. Very interesting. Here we use our reverse transcriptase on the RNA of cells, and this gives us cDNA fragments, which we can turn into oligos and label, and then hybridize to various positions, various spots on our array, and find out what genes are being expressed, whether these genes have, certain of these genes have particular mutations of interest. So in that way, it's a very powerful tool. The final one I'm going to talk about is one that you probably have actually ordered at some point if you've been in the NICU, which is the chromosomal microarrays. Works on the same system, the patient labeled material labeled green and the probe material labeled red. What this does is it just goes down and it finds little probes, oligos usually, for positions close to each other on every chromosome, so that the slide is just a sort of a record, starting with chromosome 1 and ending with chromosome Y, and going all the way down to the end. The company we use that provides this service has about 180,000 different probes on their CMA. So, and if it happens that you have a duplication, you'll get a green signal as the 3 copies overwhelm the 2 copies on the probe dot. If there's a deletion, you'll get a red signal because there'll only be 1 copy of the DNA from the patient and 2 copies on the probe. This is a terrific advance and gives much higher resolution for these sorts of deletions and duplications than we're able to do with cytogenetics. Now, in our own lab, we do a number of tests. We do a whole bunch of PCR tests, one for a thrombophilia gene, some for cancer genes. We test viruses, like viral loads, in fact, and the existence of the virus. And then we do fish also, mostly for cancer operations. In doing this, I'd like to talk about the specimens required and the stages involved, and something about the controls necessary for accuracy. So let's do the thrombophilia first. So this is going to find factor V Leiden, factor 2, the methylene tetrahydrofolate reductase gene, and so forth. So, okay, so there's several mutations that it looks for, and I'd like to draw your attention to some technical advances that are here. Instead of having your typical array where you use fluorescence technology, this type of technology is based on microelectronics. Your probe, the spot with your probe on it, is actually a kind of electrode, and you're going to hybridize your patient DNA with a little ferric label oligonucleotide. So what happens when you get a match here, and this particular patient sample hybridizes with this particular spot, it's got this ferric label bound to it also, and when voltage is swept over it, it makes a little report. This would be the background signal for this site when the voltage is swept over it, but this little curve here tells you that there's something going on there, that there was hybridization at that site. And so what you can do here is you just make your DNA single-stranded with a special exonuclease digestion. You attach your reporter ferric oligonucleotide to your little clone sequences. After all this is done after PCR, then you just insert this into a cartridge, and a cartridge does everything for you, and you can get it'll make judgments about the signals that you're getting. This would be a good signal. This would be a poor signal, which you would drop out. It can look at the shape of the signal to tell you whether you've got a homozygous or heterozygous signal coming off of that particular spot for that particular allele. Or if it's somewhere in between, it'll just drop it as indeterminate and won't count it. And there are all sorts of controls that we run with this to make sure these results are accurate. So, and that's true of all of these molecular tests. It's not just a single dot that's going to give you the information. It's a dot which will give you the information you may be looking at, and a couple of other dots beside it which want to disprove that. So in good scientific matter, we try to keep things well controlled. Now, here's another test for the epidermal growth factor receptor mutations. Happens in small cell lung carcinoma. We're looking for several mutations here clustered around exon 18 to exon 21. And this is done by, it's a qualitative real-time PCR test. So we've got, different patients here. And the outcome of this test is actually quite important, because if you have a selected mutation from the 8 or so mutations that we look for, you could be a candidate for a drug. There are 2 drugs which seem to be very important as kinase inhibitors found in mutations in exons 19 and 21. So this is the way we then use the molecular test to try to find which is the appropriate therapy. It has an interesting workflow in that it starts off not from peripheral blood or bone marrow or anything like that, but from a paraffin-embedded formalin-fixed specimen. And you stain that, find out where your tumor is, use the adjacent unstained piece of the block to extract the DNA. That gets counted, set up the real-time PCR. You go through the automated analysis and get those results that I showed you. Now, this is an interesting one from a cytogenetic point of view, because we all remember the Philadelphia chromosome. This little chromosome that results from a translocation between the Abel leukemia virus on chromosome 9 and the breakpoint cluster region in chromosome 22. It produces a fusion protein, which is a thymidine kinase, and this causes cellular proliferation and sets you up for chronic myeloid leukemia. Well, now we have this wonderful drug. The first one was imatinib, and it has some descendants, which are very effective treatments for this, because this little drug will sit itself in the active site of this fusion protein enzyme and basically slow the disease down. Well, we may want to know in patients how well the drug is working, okay? So we can now do a kind of PCR analysis for this, in which we isolate the mRNA from the patient's peripheral blood. We do a cDNA change, and then we do real-time PCR with a TaqMan fluorescence reporting. And what this does is it tells us what the progress of this reaction is. For example, we want to know how much of that mRNA is still active in the disease state. So here's a patient coming along, and at some point it goes into the exponential phase. We know that that patient then still has enough of the mRNA, which is not being taken care of by the drug, okay? So we'll want to change his drug regimen. This patient doesn't seem to break up into the exponential phase, and so we know that he's going along pretty well. Okay, so here again is the use of a technique to assess the patient's status. Now, for the HIV-1 viral load test, that's very interesting. Get the virus particles isolated from plasma. You release you lyse the virus. You get the RNA out, and it serves as a template for a special enzyme that both does reverse transcription and acts as a DNA polymerase at the same time. Scientists are always looking for very interesting natural phenomena, curious sorts of polymerases that occur in various species that they can use that have odd properties that let you do things like this. So then they found it in this case. So, and just as a control, an HIV-like control sequence is added to the RNA just to make sure that the reactions are proper. Real-time PCR amplification and automated analysis, and we come out with a report for viral load. Now, in clinical genetics, which is closer to my field, here are a number of familiar tests, all of which we do, and we are familiar with the tests and the specimens needed for these tests. This may be unfamiliar to you, but I'll talk about that in just a second. This is, in fact, the unfamiliar test. It's a new maternal screening test, which is done with cell-free DNA circulating in the mother's serum. The cell-free DNA is from both mother and fetal sources, and we can see it beginning about 7 weeks of gestation. And the trick is to amplify selected sources from the cell-free DNA, which should give you sort of like histograms proportionate into the lengths in the DNA content of the different chromosomes. So this is what you expect to see after you do that for chromosome 1. And this is chromosome 21, which is just a small chromosome. That's what you expect to see after isolating and amplifying that DNA. But if this chromosome 21 looked a whole lot larger, then that would be an indication that there is a trisomy 21 in the fetus. Okay? Because you assume there's not trisomy 21 in the mother. All right. So it's a very good test. It has a very high negative predictive value, which means that if you're found negative, you're in pretty good shape. The positive predictive value is also pretty good, but you need to have a more invasive test like amniocentesis after that to confirm that result. But this is a new kid on the block, and not many insurance companies are providing for it because it's expensive, but it's probably going to become a standard sometime in the future. So when we do a genetic evaluation, sometimes the syndrome is obvious, sometimes it is not at all obvious, sometimes it's not even clear whether it's genetic, sometimes if it's genetic, we don't know whether it's Mendelian Chromosomal due to some sort of complex inheritance. And so we have our various tests that we do. Usually the new microarray technology is very much important. But then, of course, there's a new technology, which is on the block now, and I'll talk about that later. But one of the dilemmas you come up with in your clinical work, if you're working in the NICU in particular, do we do chromosomes on this baby, or do we do the chromosomal microarray? Well, the microarray is more expensive. The chromosomes can be simpler. Sometimes it's obvious it's a chromosomal abnormality. We don't really need to do the chromosomal microarray. But I just throw this up here. You can't read it because the print's too small for you. But the American College of Medical Genetics has produced an algorithm for doing this making this kind of decision about chromosomes or chromosomal microarrays. And if you need a copy, come up to me and I'll get a copy to you. Now, about chromosomal microarrays, they cannot detect the point mutation problems, the small mutations. They are better at detecting the larger scale mutations, although once they detect it, like a missing sequence or something like that, or a sequence in excess, they can tell you all the genes that are in that sequence, so it's really cool. And they can go to a level of resolution much better than a cytogenetics test can do. Now the next big thing is the next generation sequencing. And this is not done like Sanger sequencing, where you have to do one clone at a time. This is done with massive parallel sequencing, which you do thousands, hundreds of thousands of clones at the same time. Now you can do a person's genome in an afternoon with this technique. It's highly computerized and somewhat expensive to do. But there are two major forms for this. You can do a whole genome sequencing, which is you just sequence everything in the genome, or you can do a targeted kind of sequencing where you do just the exons, that is the active genes. And those constitute less than 2% of the genome, so that's a big shortcut, and you can get a lot of studies done if you just do the whole exome sequencing. Okay, it happens that whole exome sequencing is very powerful for discovering genes in rare Mendelian conditions. So that they did a study of 362 families with rare conditions, a whole variety of these rare conditions. And the whole exome sequencing found that in 105 patients had a known gene. So it was good to do the diagnosis, but the gene had already been known to cause this particular condition. Then 83 had a previously unknown causative gene. That's really remarkable that they could pull this out of just studying the family, just one family, to be able to find the causative gene. There were 28 candidate genes. Genes that look pretty good. You need further investigation to study. 146 of this 362 failed to reveal even candidate genes, so that's a real mysterious thing. The lesion may lie outside the exons, and you may have to do whole genome sequencing to find that. But this is a novel gene discovery rate of 23%. That's really remarkable when you compare past results where these various conventional tests like karyotyping and looking for the MCP2 in girls for the Rett syndrome and things like that. The yields on these tests done individually, not as a multiplex testing, are in fact rather low. So the whole exome sequencing is really amazing, particularly in these cases where you have a family where children have a rare disease. They undergo a diagnostic odyssey where gene by gene people look for what might be causing the problem. And you have the innate difficulty in that your sample size in this very rare disease is maybe like 1. You don't have any other comparisons to make. The biochemical defect is not known. So the next generation sequencing technique, particularly the whole exome sequencing to start off with, may be able to find that disease causative gene, and you can give it a name and end the diagnostic odyssey. Now, now we have available a whole variety of tests. These would have to be done on a referral basis because we don't do them here. But look at what we could do the single individual tests if we know what we're looking for. The multiplex testing, however, gene panels by disease is the next thing that we should go to if you don't know what you're looking at. So Baylor, for example, has what they call their cardiovascular panels. And you can have particular diseases like Marfan's, where they'll do 20 genes, or larger subject like congenital heart disease, where they have 117 genes they can look at. So if you have a congenital heart disease baby, and you think you know what you're looking at, this could make it even more precise, okay? You have long QT syndrome. They could look for all the 15 genes that are involved in that. And there are 21 other panels that are part of this cardiovascular panel. Mayo, likewise, has the neuromuscular panels, which look like they've got about 21 different panels there. And Mayo has set up this algorithm, a testing algorithm. You start off, you wonder whether, if you can pretty well tell whether the kid has spinal muscular atrophy or the spinal bulbar muscular atrophy, or certainly frontotemporal dementia, or ALS. And so you segregate your patients that way. There's a small number of genes you test for, but if those results are negative, then you can reflex to a panel of genes that has 17 genes in it that can tell you maybe you can pull your disease out from that. Or if this is negative, then you have to consider the whole exome sequencing or even the whole genome sequencing. So you can see how it goes from very specific to very general in the search. So I think we'll, we're running short of time here. I had a little thing to run through here, but it's a patient with leukocytosis and how you might arrive and what kind of tests that you would use. But just to summarize, since time is short, we've got tests that look for gene products versus nucleic acid base. That's our first historical revolution there. We went from the gene products to the nucleic acids themselves in our testing. And we did PCR versus microarray versus next generation sequencing now. We've got targeted tests all along, and we can even do targeted tests in the microarray context, as I've just showed you, which are global tests where we just don't know what it is, and we say test everything, and we're agnostic about what the disease is. Nucleic acid tests, single target versus multiple and multiplex tests, panels for selected genes, which we just went through, whole exome and whole genome sequencing. All these things are now available to us in that order. Any questions? Okay, I regret that I wasn't able to get into other topics here, but time is limited. All right, thank you, Lynn.